the survival of animals and plants and humans. Just like animals survive, the survival of plants is also dependent on their ability to respond to changes in their environment, and this maximizes their survival chances. For example given, growing towards light maximizes the rate of photosynthesis and therefore glucose production. And of course, that's because of photosynthesis. So this is one thing that helped it to survive, okay? Other than that, producing harmful or foul-tasting chemicals in response to being eaten by a herbivore reduces the likelihood of being eaten. Now it can survive more. Producing flowers at the right time of the year increases the chances of reproducing successfully. Alright, so now we've mentioned 3 points. First point we mentioned here, that's related to photosynthesis, that if it grows near light, it can survive. And if it produces foul tasting chemicals or harmful chemicals when being eaten, this leads to it not being eaten anymore. It reduces the likelihood of it being eaten. Also, for the flowers, if they are grown at the right time of the year, this increases the chances of reproducing successfully. Plants can respond to several types of stimuli, light and gravity, and these are stuff that we already knew before. The light and gravity, the physical objects, the herbivory, water, and physical touch. So we've known before light and gravity. Other than that, we have being eaten, that's one thing we said before. Physical objects, touching it, or we're gonna know more. Herbivory, water, and physical touch. Okay, so to memorize it easier, we're gonna summarize it into some compartments. The first part is light and gravity, and that's what we know in the previous biology O level. Light and gravity using oxygen. We've known the physical objects and physical touch. Physical, that's a good way of memorization. Physical touch. And we've got the herbivory, which is being eaten by herbivores, and water. Now that's food and drink, physical objects and physical touch, light and gravity. Okay. Unlike many animals, the plants do not possess a nervous system. The responses of plants rely on chemical substances that are released or altered in response to a stimulus. So, unlike we have many animals in the world, and these animals possess a nervous system with a brain. Okay, and brain and the spinal cord and the CNS and the PNS, all these. However, the plants don't possess a nervous system, but they can respond to stimuli by releasing chemical substances or altering them in response to a stimulus. First part in here is the phytochrome. The phytochromes are plant pigments that react to different types of light, leading to different plant responses. Phytochrome pigments exist in 2 forms. We have the PR, which is the inactive form of phytochrome, and it absorbs light from the red part of the spectrum. Okay, we have a red part of the spectrum and we have the far red part of the spectrum. The PR is the inactive, while the PFR is the active. Okay, so that's the inactive and that's the active form. We've said that the inactive form absorbs light from the red part. You can say that it absorbs it from the red part because, as you can see, it's phytochrome R, which is red, and here it's phytochrome far red. Okay, And here it's R, it's red only, it's inactive. It's FR, it's active. Alright? And the inactive form is little, more little. The wavelength is a smaller number than the 730 nanometers, which means it's a less word, which is only one letter, though it's R, which means it's inactive, okay? Absorption of different wavelengths of light causes a reversible conversion between the phytochrome R and the phytochrome FR forms of phytochrome, okay? So different wavelengths of light leads to a conversion. This is a new thing. If I have different wavelengths of light that's shown into it, different wavelengths of light, it causes a reversible conversion. There is a conversion and it is reversible. Okay? When the PR absorbs red light, it is converted to PFR. And when the PFR absorbs far red light, it's converted back into PR. However, in absence of red light, if we don't already have red light, the unstable PFR gradually converts back to PR. So PR already absorbs the red light, and whenever it absorbs it, it's converted into PFR. While the PFR absorbs the far red light and is converted to PR. And even if there is no red light, the PFR, which is unstable, is gonna be gradually converted at the end of the day into PR. So we can see how it leads to PR at the end in here. Phytochrome and germination. Early observations of light exposure and seed germination showed that seeds exposed to red light would germinate, while seeds exposed to far red light not. Why is that, however? We said the red light now would make it germinate, which is a lower wavelength, and now the seeds exposed to the far red light would not. Let's know now. It is now thought that phytochrome is responsible for this observation. Exposure to even a short burst of red light, if we have many little red light, it converts the PR into PFR, and this triggers germination in seeds. However, the far red light causes the PFR to be converted back to PR, and this reverses the effect of any red light exposure and prevents germination. You might think it's pretty complicated, and it might be actually, but we're gonna simplify it together. Let me see in here. I'm sorry. Okay. So we said that if there is an exposure to the red light, this leads to germination. But if there is far red light, it would not. However, only the far red phytochromes are the ones that lead to germination. But why? Because it's like opposites. They are opposing each other. If I have a PR and it's exposed to red light, this means it's converted to PFR, which wouldn't. It wouldn't lead to germination, or would it lead to? It would trigger germination. Okay, so it all depends on the type of exposure. If it's exposed to red light, then it would be converted into PFR and it would germinate. But if it was exposed to the far red light, which means it's going to be converted into PR, and this means no germination. You might get lost, I know. Phytochrome and germination. So which phytochrome eventually leads to germination? It's the PFR. And by the use of what? By the use of red light. PFR and red light. PFR and red light. I want you guys to memorize it because it's quite hard to remember. But like the phytochrome and germination, all the thing is about red lights. If you mastered knowing that red light causes it, that exposed to red light germinate while the far red light won't. And this is because the red light would convert the PR into PFR, and this triggers germination. And the far red light causes the PFR to be converted back to PR, which reverses the effects of any red light exposure. And we love red lights. We love red lights. Okay? Remember it. Valentine's Day. Red light is better than the far red. We don't need no far red. Just keep it early and close. Valentine's Day, red light. Okay? Because it converts the PR into PFR, and that doesn't reverse the effects of any red light exposure. Okay? We hate the far red lights. Do you want to date me now or date me later? If you love me, you will love to date me now. Yeah, okay. Stop going off topic and let's continue. I'm not gonna go off topic anymore. Now let's see this part, phytochrome and flowering. Flowering in plants is controlled by the stimulus of night length. Now we need night. Alright? Nights are shorter during the spring and summer and longer in the autumn and winter. Yeah, physics information. Some plants flower when nights are short, and some flower when nights are long. When the nights reach a certain length, genes that control flowering may be switched on or off. Isn't that crazy? It is. Okay, when they reach a certain length, the genes might be switched on or off, and this leads to the activation or inhibition of flowering, because of course genes are responsible for flowering. Activation or inhibition of flowering. Genes that are switched on are expressed. The genes that we switched on are expressed, and this leads to the production of the polypeptides for which they code, while genes that are switched off are not expressed, so no polypeptides for which they code are not produced, and there is no flowering. The flowering here now, however, depends on the polypeptides, which could only be formed if the genes are expressed and they are switched on. The length of light can be detected by a plant because it determines the quantities of different forms of a pigment called phytochrome in the leaf. Okay, so how can just a simple leaf like that, this is our leaf, and damn, our leaf actually knows how long night is. Okay, it knows the time. Isn't that a little bit crazy? Not a little bit, but very crazy. I'm gonna tell you why though, guys. Because it can determine that not from looking at the clock, but from determining our pigment in here, which is called phytochrome. It can determine the phytochrome levels because during the day, levels of the PFR rise. Sunlight contains more wavelengths at 660 nanometers than 730, so the conversion from PR to PFR occurs more rapidly in the daytime than the conversion from PFR to PR. Okay? During the night, levels of the PR rise. So when they know that the phytochrome R rises, this means that there is more night. Now it knows that we have more night today. I'm gonna decide whether I'm gonna flower or not. And this depends on different types of flowers. Red light wavelengths are not available in the darkness, and PFR converts slowly back to PR. Why? Because we said that we have unstable PFR, which will be turned into PR directly if we don't have the far red lights. Alright? And I want to help you guys memorize this part. So I don't know, maybe you can think of it as the phytochrome in far red light. The far red, which means you have many lights. Many light, we're in the day. Okay, that's far red. However, this is red. Okay, not very high level of red. We are at night. And we have a diagram here, and you can see our pretty little red rose. Let us zoom in. The PR levels rise during the night and in the darkness. And this is the PR, which is inactive. The PR, of course, is inactive while the PFR is active during the day. And we said that before. So the PFR, which is in here active during the day, can only be reached if we had red light to convert from PR into PFR. But if we had far red light, that's not a good thing because it's gonna get back the PR. You know what I mean, everybody? In dark and night, it is slow. Okay? Now our beautiful, active, amazing PFR. Far red light and flower. FF though. I know we've made many acronyms, but that's only to help you guys memorize and understand as well while relating to the information. We have that PFR, which will bind to receptors that either activate or inhibit flowering. So this is our boss in the story, the PFR. This decides everything. And here is a summary. Of course, we said that the PR is converted to PFR. That's why we love nights. Why do we love nights? Nights have the red light, which is at a low wavelength of light. Okay, it's at a low wavelength of light. That's why whenever the red light is available, it can convert the PR, which we don't like. It's inactive, using red light into PFR, the one we like, and it's active and beautiful. In long day, we have the long day plants. They flower when the nights are short, like in summer. When nights are short, the day length is longer. Yeah, makes sense. They are long day plants. They have more morning. So that's when they flower. In long day plants, high levels of the active form of phytochrome activate flowering. And why do you think so? Why is there high levels of the active form of phytochrome that activates flowering? We have a high level because we have in short night that PR is converted into PFR. You know what I mean, everybody? The flowering occurs due to the following process. Days are long, so PR is converted to PFR at a greater rate than PFR is converted to PR. Therefore, the active form of the phytochrome, which is the PFR light, is present at high levels. And these high levels of the PFR, which is the active, beautiful one, activate flowering. It activates expression of genes that stimulate the flowering. It is thought that the PFR acts as a transcription factor, where the active gene is transcribed and translated, and the resulting protein causes flowers to be produced rather than stems and leaves. And that's how flowering takes place. We don't get the stems and leaves. They are not in this phase. We're in flowering, and we have the flowers to be produced. And this is all because of the PFR, the flower-inducing far red light phytochrome. Now let's move to the phytochrome and transcription. Okay, evidence suggests that in addition to the role in germination and flowering, they are also involved in plant responses to light and gravity. Phytochromes are thought to influence gene expression in plants by acting as transcription factors. Okay? So now the phytochromes, which we have the two types, PR and PFR, act as transcription factors. The PR is converted to PFR in the presence of red light. PFR can move into the nucleus via the nuclear pores and binds to the protein in the nucleus known as a phytochrome interacting factor 3. Phytochrome interacting factor 3. Alright, so when we have the PF, which is our active form, it's gonna enter the nucleus via the nuclear pores and binds to a protein in the nucleus which is called the phytochrome interacting factor 3. Phytochrome, I want to write it to you guys again. Phytochrome interacting factor 3. Phytochrome interacting factor 3, which is the PIF3. Once bound to PFR, the PI factor 3 initiates transcription. It is thought that the PFR and the PIF3 together are able to activate various different genes and also control many different aspects of the plant growth and development. Alright, so they both can activate different genes acting as the transcription factor, and they control many aspects of the plant growth and development.